We're on S61. We're talking about the photosynthesis lab. Y'all remember this thing? What we did was we cut out leaf discs. Y'all remember this?
45 RPM.
And we put the leaf discs. Well, first thing we do with the leaf discs is we put them in a syringe. And what do we do with the syringe? Do y'all remember that?
Turn it into a vacuum.
Why do we do that?
Giving oxygen out. And they sink.
We take a leaf, we punch a bunch of holes in it, we put the leaf discs in the syringe, pull up on the syringe, and that creates a pressure that pulls the air out of the little leaf discs. So there's no air in them. Then, I feel like no one's listening.
I'm listening.
I feel like one person's listening. Then, if you put the leaf discs under water,
They're going to sink. There's no oxygen in them.
they're going to sink. And then, if you shine a light on them, or just make sure they're in the light, they'll do photosynthesis, you see. And as they do photosynthesis, they make oxygen as a byproduct. Y'all remember the photosynthesis equation?
C6. 6O2 plus 6H2O. 6 CO2.
6 CO2 plus 6 H2O
Plus light.
yields glucose plus O2.
6O2.
Yeah. And glucose is C6H12O6.
Can we move to the
This guy?
Yeah. How can his head?
We took it off for a test. You have to put it back on. Okay, so they're doing photosynthesis. The whole purpose of photosynthesis is to make food, by the way. And y'all remember, this is kind of a good review here. Y'all remember from the carbon cycle, this is how carbon dioxide is pulled out of the air and into the tree. This is how we say carbon dioxide is fixed through a process of photosynthesis. It comes out of the air and is made into glucose, which is a solid substance in the tree. And that's why growing trees reverses global warming, because it pulls in that carbon dioxide from the atmosphere. Carbon dioxide is no longer floating around in the atmosphere. It's a solid form inside the tree. It's not going to be trapping any heat and won't be making the earth warm. So that's kind of an important extra thing about photosynthesis. Anywho, as you shine the light on these little leaf discs, they will be doing photosynthesis and making oxygen as a byproduct, and the oxygen builds up inside the little leaf discs, and the leaf discs will then float to the top. And so the faster you can time how long it takes them to float to the top, and the faster they float to the top, the faster they're doing photosynthesis. So you can make this into an experiment. You can, for instance, didn't you do different colors of light? He shined red light, blue light, and green light, and saw which color light makes him do photosynthesis the fastest. What color?
Yellow.
Yellow did it faster. Did yellow do it faster than white? Did you do white?
White was clear. Obviously was fastest.
Clear was fastest, and then yellow was next. Did anyone else remember their experiment? What you did? Yeah, what you do?
Different temperatures of water.
You made the water different temperatures. Good. And did it rise faster in the hotter temperatures? Very nice. You see? They might ask you some sort of derivation on that experiment. However, we noticed that they didn't rise at all when? We did a little experiment and we saw that they didn't rise at all if you didn't have something in the water.
Carbon dioxide.
Yeah, well, we put bicarbonate, a bicarbonate ion, which is HCO3. If we put bicarbonate in the water, they rose. But if the bicarbonate was not in the water, they didn't rise. And why is that?
They need carbon dioxide.
They need CO2. They got the water. They're in the water, but they need CO2. And there's not enough CO2 underwater for those things to use, so you got to put some in there, and that's the form. The HCO3 will dissociate, and CO2 will come out of that molecule and can be used by the leaf.
Wait, they wouldn't rise if it was in there?
If there was no HCO3 in there, they wouldn't rise. And we did an experiment, Cole, if you remember, where there was no HCO3 in there, and none of them rose. Do y'all remember that? Is this exciting, Dante?
Sort of.
You don't think it's exciting, I think.
So wait, HCO,
He's honest.
At least he's honest.
He's honest.
HCO3 the bicarbonate?
Yes. That's we just put it's baking soda.
It's also known as sodium bicarbonate. Is that different?
Yeah, actually, I think it does have sodium, NaHCO3 or something like that. But we know it has the CO2 molecule in there somewhere. Okay? I'm just trying to refresh your memory on these. I know you should still, for the lab test that's Monday, you should still study this stuff. Go back over it, look back over what you did in your logbook, read over what it says in the lab book.
Are we going over every single one?
Every lab? I'm doing that right now. Yep. It's good, isn't it? Yes.
What do we do in the lab test, like the lab exam on Monday?
It's a multiple choice and essay test, just like any
So
other
we don't-
test, but it's
we don't
just
do
over
it.
the labs. No, you're not going to be in the lab room. Just over the labs.
You ever taken a lab?
No. All right? All right. I'm going to be gone, but you'll have a sub here that's giving you the test. We got region golf that day. Okay, so the next one was respiration, which is on S71. And the lab book talks about a whole big long lab where you're using like a syringe and you call it a respirometer. But we didn't do ours this way. In a respirometer, you put little tiny peas or beans or some sort of little organism. You could even put crickets in there. And if you dunk this whole thing underwater, you will see the water level as these things do their respiration. Do y'all remember the respiration equation?
6O2 plus C6H.
How can I change this to the respiration equation?
Put the air the other way.
That's respiration. Food and oxygen form these waste products. And the important thing that forms is what?
ATP.
ATP, energy, which is used to make ATP. So, respiration is the process, going this way, is the process of breaking down food to make ATP. And water and carbon dioxide are produced as waste products. Glucose is food. And look, you have to use oxygen to do it efficiently. Do y'all remember what organelle in the cell does this process?
Mitochondria.
The mitochondria does most of that process. A little bit of it is done outside the mitochondrion in a process we call glycolysis, which is an anaerobic process. Glycolysis does not use oxygen and makes a little bit of ATP. But the rest of the process is done in the mitochondrion and uses oxygen to break down that glucose fully. And that's where most of your ATP gets produced. One glucose molecule will make about 36 ATP, something like that. as it's being broken down. And those ATPs can be used in your cells to do all sorts of things. One candy bar has about 10 to the 20th of those molecules in it. I mean, it's a huge number. So you're making a lot of ATP every time you eat. So, any evidence coming out of your body right now that you are doing this process inside you?
Heat.
Yeah. Well,
Releasing carbon
there is
dioxide.
some heat. This isn't a perfectly 100% energy transfer.
Carbon dioxide?
Carbon dioxide. You're breathing it out.
And we breathe out a little bit of water, don't we?
Yeah, some of the water comes out too, you're right.
Well, we drink most of our water, right? You know,
Yeah, you drink water to replace it.
'cause 6
So,
is really not bad.
in this experiment, if you were using a respirometer, which is a device that looks like this with a pipette on the end, and what you do with the respirometer, now we didn't do this, we did it a different way. I'll talk about the way we did it. But you put some peas or beans in here, and as they do respiration, they're taking up oxygen. And they're putting out carbon dioxide, but if you put a little chemical in here, we didn't do it, but the chemical's called KOH. You put a little KOH in here, KOH absorbs carbon dioxide,
What does KOH stand for?
potassium hydroxide. And potassium hydroxide absorbs CO2. So think about this. Y'all didn't do this, but you should be able to see what's going on here. If the little peas or crickets or beans are doing respiration and producing CO2, taking in oxygen, but the KOH is absorbing the CO2, what would happen if we took this whole contraption and we put it under water? Put it in a big beaker. What's going to happen as these little guys do respiration and take up oxygen from the area and spit out CO2, but the CO2 is absorbed by the KOH?
Water's going to go in.
Water's going to go in there. There's a hole at the end. And water will go in there. Why do you say water will go in there, Isaac? You're right.
Because the volume inside is decreasing.
The volume of gas in here is decreasing because we're pulling in oxygen, we're putting out CO2, but the CO2 is automatically used up. So as these things pull in oxygen, the water would creep in.
How does that work?
And that's how most people in the country do this experiment. What were you going to say?
How does KOH just magically get rid of the CO2?
It's a chemical reaction. The CO2 binds with KOH and forms a new substance that's a solid, not a gas. And solids take up hardly any room, and gases take up a lot of room. Y'all see what's going on here?
So can you measure that? How much or how fast the water?
Yeah, how fast the water comes in will tell you how fast these things are respiring. And you can do the same sort of experiment that we did with the photosynthesis. You could use different temperatures of water and see where respiration occurs fastest. What do you think it would be? Where would respiration occur fastest with these guys? Hot, cold, or medium water?
Hot.
Medium.
Why do you say medium?
It's not too hot to denature proteins and all.
I guess if it was too hot, it would be nature.
But
But as you get warmer, these guys are cold-blooded creatures. They do stuff faster. So actually, respiration increases the hotter you get up to a point. Now we did this experiment with our probes. Do you remember? We had a little tube, and the tube kind of looked like this. We put the peas in there. Don't y'all remember doing this?
Yeah, that was a good one.
We stuck the probe in there, and the probe just gives us a reading of the CO2 level. or the O2 level, depending on what kind of probe you were using. And the probe would tell you what would happen to the CO2 levels. Don't shout it out. I'm going to call on someone. What would happen to the CO2 level on this probe as these guys in here were doing respiration? Sarah?
It would. So is it?
These guys are doing respiration. What's the CO2 reading going to be on the probe? What's it going to do? It's going to stay the same? It's going to go up? It's going to go down?
Increase.
It's going to increase. Because they're doing respiration, they're producing CO2. What would happen to the O2 level on the probe, Elena? It would decrease. Because they're taking up O2 to do their respiration. And every organism does respiration. You could put a mouse in here. You could put a human in there if the human was small enough or the chamber was large enough.
Now, if it was colder, wouldn't a warm-blooded animal respirate faster?
Warm-blooded animals actually speed up their respiration in the cold because they start shivering and shaking, doing more work to keep their body temperature correct. So, warm-blooded and cold-blooded animals are a little bit different.
And do plants make their own heat when they do respiration? Are they warmer than the environment, or?
Plants make a little bit of heat, but not much.
Yeah.
All animals all organisms make a little bit of heat.
Yeah.
The larger the organism, the more heat it usually makes. And some animals try to keep in that heat, so they keep a high level of metabolism going. That means they can be more active.
I see.
So that was a fun little respiration lab. Does it make sense?
Yes.
Okay.
This is fun.
We're lucky that we have those probes. A lot of schools don't have that, and it's really hard to set up those respirometers.
So on the lab test or whatever, should we? Can we? If we had an essay about that, would we do it the respirometer way or with the probe?
They'll probably set it up for you in the question, tell you which way to do it.
Okay.
They would probably allow you to use a probe if they just said, do an experiment that checks
Wait,
respiration.
is this part of the national?
Mm-hmm.
The lab exam?
Well, no. The national exam's just something you take that one Monday.
Yeah, so
The lab exam is for my grade.
You created.
Mm-hmm. Yeah.
And the lab exam goes into our final exam grade, right? Or is it?
I haven't decided that yet.
Okay.
Our actual final
Is that what y'all want?
Next Thursday, Friday.
Oh,
Final exam is Thursday and Friday during class.
I think.
And maybe Monday if the lab exam goes into it too.
Is it the next Monday, the national?
The next Monday is the national. Would you rather have the lab exam be just worth the test or be part of the final?
Part of the final.
I think
Really?
a test would be
Test.
better.
Test?
I don't really care. Whatever is good. I mean, if you
Which would help my
did
grade
better
on the
helps
final
my
and
student.
did it on the lab exam, then.
How have people
the
done-
final would-
How are people done typically on the lab test?
About like the other test?
Well, I mean, if this, since, we just had our half test, that was like our last test, but we could have another one,
I think I'd
one
rather
last.
do a test.
It'd be one last test. You could raise your test grade. I don't know.
Yeah.
Let me think about it some more. Let me ponder. I think it'll be worth about the same either way. So, okay. It would spread your exam out over more days. Maybe that's less stressful for you guys if it was part of the exam. The Lab 7, we didn't really do in its entirety. We did some of it. Do y'all remember looking at onion root tips?
Yes.
Yeah. Okay, that's part of Lab 7. We didn't do all of Lab 7, but we looked at the onion root tips under the microscope, and we saw, we're looking at this part of an onion root tip. This is kind of the area of cell division. And then if you look up close, this is the tip of a root of an onion. These cells right here are, a lot of them are in mitosis. And they're undergoing cell division rapidly. Why are they doing that there?
The root's getting longer.
The root grows and grows down into the ground. And for the root to grow and get longer, these cells have to multiply. And that makes the root get even bigger, you see? And so that's a good area to look to find cells that are in mitosis. Now, if we were to do the lab in its entirety, here's what we would have done. We would have taken an onion, a normal onion, and looked at its cells. And then we would have taken another onion. This would be our normal. And this would be our lectin onion. Lectin treated. And if you treat an onion with this chemical that's called lectin, the lectin makes the cells divide a lot more rapidly. It's a chemical that's often put on plants to kill them. Because what happens is that the root grows too rapidly and gets real thin and weak, and it ends up in the plant ends up not being able to do its job, and the plant dies. That's how a weed killer, a popular weed killer that's sprayed on weeds in your yard works. That weed killer is called, I can't remember the name of it now.
Roundup?
No, not Roundup. It's one that you can spray on St. Augustine grass, and it just kills the broadleaf weeds. It's like something 40 or something like that. Anyway, the point is, in the lectin-treated onion root, where the cells are dividing a lot faster, what do you think you would see under the microscope? What would be the difference in the way this looked to that looked?
Cells in the lectin, there would be more cells undergoing mitosis.
More cells undergoing mitosis in the lectin-treated slide. And what it has you do in that experiment is you count the number of cells and you make a little table. Cells in mitosis. Cells in interphase. And maybe we have 40 cells in mitosis and 190 in interphase here. And over here we have 73 cells in mitosis and 110 in interphase. See how this has a lot more in mitosis? Then what it asks you to do, can you guess what it asks you to do with these numbers? A certain type of test we've recently studied. Chi-square test, very good. And it asks you to say, are these two sets of numbers the same? Or has the lectin actually had an effect? This is what we would expect to see if the lectin had no effect, we would expect to see this. But this is what we observed. So this is the observed, this is the expected. Is the observed minus the expected, is this different enough to say that the lectin is actually having an effect, that these numbers are different than these numbers, not due to random chance, but due to the lectin? So you run a chi-square test on that. And the problem is the lectin is expensive, and it takes a long time to grow these onions, and it's just kind of a time-intensive lab. We didn't have time to do all the labs, so that's one of the ones I kind of skipped.
And you had the slides with the menu.
We had the slides, the regular slides, but I don't have any lectin-treated slides. I'm going to get some for next year, but y'all missed out on that. I got some coming for these workshops I'm doing this summer.
You want our books out there?
What's that?
Like, do you want these?
No, those are yours. Those are yours. You don't want it?
I mean, after the exam, I won't really need it.
Well, you can give it back.
Alright.
Did you write it?
No.
Oh, sure. Yeah, give it back. That'll be, or you could sell it to somebody. Maybe make a little cash. All right. How's that lab?
Yeah.
Lab 8. The bacterial transformation. Oh, this was fun.
It's a hard one.
Oh, was that with the auger and all that?
Yeah.
Yeah.
Let's review what we did there. We took a couple test tubes. This confused a lot of y'all, so pay attention here if you weren't too sure what's going on. Why is this box here? One test tube we called the plus test tube, and another we called the minus test tube.
What is the topic of this one?
This is bacterial transformation, page S97. We put E. coli, little bacteria, in here. I just put them as dyes. E. coli. E. coli. No?
Ricola.
Nothing there? Ricola, yeah. Okay, thank you. A couple of laughs. Along with the E. coli, in the plus tube, we also put a plasmid. Do you remember, Caitlin, what a plasmid is? No?
DNA.
Let's try Steph. What's a plasmid? A plasmid is a ring of DNA. Ring of DNA. And that ring of DNA has information on it. DNA carries information, right? And so this plasmid has info for ampicillin resistance. That means there's instructions on there that tells this E. coli to make a protein that helps them resist ampicillin. I don't know how that protein does it, but somehow when that E. coli makes that protein, ampicillin doesn't bother that E. coli. I don't know if the protein is an enzyme that breaks down ampicillin. That would be my guess. Maybe it's something that goes in the cell wall that makes the cell wall a little tougher so ampicillin can't get in. I'm not exactly sure. But if you give that little information to that E. coli cell, it'll resist ampicillin. And ampicillin won't kill it. And now this E. coli is different than this E. coli, isn't it? See what I'm saying? Okay, so how do we know that this E. coli is different than that E. coli?
Because we tested.
We need to test it.
We test it how?
You put it on auger plates.
We put it on plates. We're going to use 4 plates here. One plate is normal agar, which the agar that we were using was called I can't remember. LB, Luriabrod. LB agar. All the plates, we used 4 plates and they all used LB agar. LB stands for Luriabrod. It's just what bacteria like to eat. So theoretically, all those plates, both bacteria should be able to grow on all 4 plates right now. But 2 of the plates, we also put ampicillin. These 2 down here. And ampicillin would kill this one. But it doesn't kill this one. Because this got the information, got the plasmid. So we simply have we put the minus bacteria, some of it on this LB agar minus plate, and some of it on the LB agar amp plate, minus place. So the minus bacteria, this E. coli that did not get the plasmid, they go on these two plates. You see, that's why we have minus there. This normal E. coli bacteria, will it grow here?
Yes.
Do y'all remember the results?
Carpet.
Caitlin's nodding her head. It grew here?
Yes.
Yes, it grew in what we call a big lawn.
Close.
Cole, what do you think? Would it grow here?
No. Because the ampicillin kills it all.
The ampicillin kills it, so this should have no growth. And indeed, that's what we saw. Do y'all remember? You should have this down in your logbooks. What about the plus bacteria? These are the ones that got the plasmid. Would they grow on normal LB agar?
Mm-hmm.
That plasmid doesn't do anything to cause them not to grow here, so that ought to be a lawn too. And we saw that it was. Would the plus bacteria grow on this one that has ampicillin?
Yes, they're all in colonies.
Ah, do it, Elena. Elena said only in colonies. Now, why is that? Why wouldn't they grow like crazy
Because
in a lawn?
Not all of them got the
took it
plasma.
up.
Not all of them were able to get the plasmid. We only put a certain number of plasmids in there. And so what you can do is you can count the number of colonies. Each colony stands for one bacteria that took the plasmid in. And overnight it grew into a colony.
That is when you turned your thing on.
When I turned the...
Or we forgot to turn the incubator.
Oh, the incubator. Yeah, I forgot to turn it on. Right, that's true. They grow faster in the incubator. Something interesting here. This colony of resistant bacteria will destroy all the ampicillin around it. And what you'll see sparking up after a few days are little daughter colonies around each one. The daughter colonies are E. coli bacteria that didn't get the plasmid. But they couldn't grow on the ampicillin. But when the ampicillin was destroyed by these bacteria, that then allowed them to start growing. So we call those little daughter colonies or satellite colonies. And if you let this experiment go too long, you'll start to see a bunch of those growing. And I think didn't we have a little bit of problem with that in ours?
Yeah.
Because I let them grow a little too long. What'd you do?
We did you know the rain thing we used to pick them up with?
Yeah.
Ours was super, super hot. It burned it.
And it killed them?
Yeah.
So you didn't get good growth?
Well, it was just one little spot.
Oh. Does this make sense? This experiment? Yes. Yep. It's a good experiment, isn't it? So we use these plasmids also in the next experiment, which we also did, the restriction analysis. Turn to page S115 and that'll probably ring a bell.
Low risk trade reaction, right?
Yeah, you need to do PCR. We didn't do PCR, but it had to be done to produce the plasmid. What we did in this experiment was we got a gel. That big square I just drew, that's a gel. And we put a bunch of wells in the gel. With the cone, do y'all remember that? And we injected DNA into our well, and we had three types of DNA. We had, now in ours we did we do a mystery?
Oh yeah. Who
Yeah.
killed this tractor?
It was Miss Bradford versus Miss Wallace.
Okay.
Bradford killed him.
So we had Bradford DNA. We had Mullis DNA.
That was Mr. Willis.
That was you.
Yeah, it was Mr. Willis.
It wasn't Bradford?
No, it was Mr.
Bradford
Willis.
in my lab book that you did it.
Okay, so
Yeah.
Bradford, Willis DNA, and who else?
Crime scene.
Yeah,
And crime scene? Now, actually, in actuality, this wasn't crime scene DNA, Bradford DNA, and Willis DNA.
What was it?
You know what this was? Russell, you okay?
Alright.
See? You alright?
I'm sorry. No.
You not alright?
No. Can
Do you need to
Step
go
up
see
a-
a nurse or something?
no, guy, just step out for a second and get some
Yeah.
something to drink. Thank you.
Yeah. Can you take over the... Just grab it.
That's it. I'll take it.
Hey.
There you go.
Closer.
Alright. In actuality, this wasn't crime scene DNA, Bradford DNA, and Willis DNA. In actuality, this was a plasmid. And this was a normal plasmid. I don't know which was which. I think I mixed them up for different groups. Normal uncut plasmid. And this was a plasmid that had been cut with a restriction enzyme called EcoR1. Do y'all know what a restriction enzyme is?
Keeps transcription occurring.
Nope.
No, that's-
Isn't restriction enzyme something that binds to a certain part of? Oh, it stops.
I still got the magnets over here. I've been too lazy to put them up the entire year.
Doesn't it?
And my laziness has finally paid off. A restriction enzyme reads the DNA sequence, and whenever it sees a certain sequence like GAA, TTC, it makes a cut in the DNA. And remember, it reads forward and backwards, so the other side it reads GAA, TTC, and so you get a cut there, a little jagged edge. Do y'all remember that? That's what a restriction enzyme does. It cuts DNA. It's like DNA scissors.
So what good does that do?
So this little plasmid right here, which is a loop of DNA, here it's uncut. But here it's been cut by a restriction enzyme. And so it's in pieces. And here, this DNA has been cut by a different restriction enzyme, which was called HIND3.
Go ahead. Do you want to take it or?
And so, if this is the normal uncut DNA, you would probably just see kind of a big band going down and not going down very far, because the bigger the piece of DNA, the shorter distance it goes through the gel. Remember, we put an electric charge here. A negative electrode on one side of the gel and a positive electrode on the other side of the gel. And the DNA is negatively charged, so it goes toward the positive electrode. Y'all remember this?
Mm-hmm.
And so if it's cut, if the plasmid has been cut a few times with EcoR1, then you'll see it's in several pieces. And the pieces go different distances. And this plasmid that's been cut with the IN3 would go in different pieces still, because it's been cut in different places. And that's why you get different looks of the DNA, because they've been cut in different places. So if you're doing it, when they're examining DNA from a crime scene, what they do is they take DNA from different people, and then they cut it with a restriction enzyme. In an actual crime, you use the same restriction enzyme on everybody's DNA, but your DNA, it'll cut in a different place than your DNA. and my DNA. And so we'll get different runs. And if you found at the crime scene, if you found DNA that was cut here, that matches this one. So Willis's DNA was at the crime scene. Around Mr. Willis. Where is he? He's with his lawyers. You're going to have to do better than that, Mr. DA. That's my little crime fantasy. I don't know. No? This doesn't seem to interest y'all? Is that all just gibberish what I was speaking there?
No, it's fascinating.
Here's what I want you to do. I got a little homework for you. It's a couple of essays that are over these labs. I want you to look at these essays. So take a sheet of paper and jot down a little outline like we do in class. Take about 10 minutes just to jot down an outline of what you would write for each one. You can use your lab books for this if you want to. Does it make sense? Make you sensey. I'll give you a grade on it.
Oh yeah, maybe.
I'll show you how they graded this. I'll show you the grading rubric so you can see how they graded.
Okay.
We done here?
I suppose so. Want me to
You can turn it off, yeah.
Okay. And